So thank you very much. Thank you, Pablo, and the organizers for this invitation. I couldn't be better positioned to speak. So Enzo, speaking after Enzo was just perfect, okay? So his talk is a brilliant talk, and he mentioned things that I'm going to mention here. So for those who are not in Brazil, we are myself, Pedro Carelli, and Evaldo Vasconcelos, we are together in this Systems Computational Neuroscience Lab, which is here in Recife. So Recife, for those who are not Brazilians, is here, for some Brazilians as well. And everything I'm going to talk about was made in collaboration with many people, including Gustavo Cambrainha, who's here, yes. And so a very, very brief introduction. So this 1963 Nobel Prize for Hodgkin-Huxley for explaining how one neuron spikes. The important thing here is the year. 63 is barely 6 decades ago. So they essentially, I think it's one of the most impressive landmarks in biophysics to be able to describe how one neuron spikes. So they proposed this for couple differential equations and explain how depolarization occurs as sodium enters and repolarizes, potassium goes out, and they're hyperpolarized. So it's a major feat of science, in my opinion. Spectacular experimental and theoretical work. And so we know this for a while, but it turns out that we have 100 billion of those guys in our brains, each one connected to 10,000 others. So these are rough numbers. So according to some, this is the most complicated object in the known universe. I don't know, but good candidate, at least one of them. So one neuron is understood to some extent, but many, like that many, it's a whole other story. So this is where many people, including ourselves, are involved with, as Enzo was giving us very wonderful examples of the kinds of things you try to do to understand collective phenomena, collective neuronal phenomena in high dimensional data. So in our group, we try to, on one hand, that's how I started. I grew up as a theoretical physicist in statistical mechanics, so we try to do models, theoretical computational models. But then I started receiving experimental data from biologists who were challenging me. Oh, you have these ideas, nice. So try to prove them with our data. And then eventually this led me to recruit Pedro Carelli, that I showed the picture before. He is an experimentalist, to try to put together an electrophysiology lab. And I was inspired by two people, essentially. So Roland Köberle in São Carlos, Brazil, and Gabriel Mindlin, my good friend. He was a major inspiration. He's such a nice friend and such a great scientist. I never said it in public, but he was one of my inspirations. And like him, I didn't do it on my own. I had this help from people who can do the experiments. So one of the ideas that have guided most of my research in the last 20 years is this critical brain hypothesis, this conjecture that the brain is understood as a dynamical system operates near a critical point. So this goes back many years. So Dante Chiavo was one of the proponents. Here are some now old reviews about the idea. So this is a cartoon of the second order phase transition in which you have some order parameter here, some control parameter, and the qualitative change of behavior continues 1 year. So the idea is that the brain would be sitting near this place. Why on earth would you expect that to be true? Because based on many models, theory predicts that at criticality, one could have things like optimal transmission capacity, optimal information processing, larger repertoire of metastable states, optimal sensitivity and dynamic range. And if I had a brain, I would love to have those things. So that's the whole rationale, the hand wave argument is that all these things are supposedly highly desirable for a brain, and therefore there will be evolutionary pressure. And then questions arise such as if the brain is critical, big if here, what is the phase transition and this kind of things. So I would try to give just a very rough a sample of ideas we explored to show how we have been applying ideas from physics to models in biology and data in biology. So we're trying to combine models and data analysis and experiments Okay, so it's a mixed bag of stuff. And Enzo just gave us a spectacular view of these ideas, showing that if you don't put your model here, it doesn't work well to fit the data. That's what he just told us, very impressively in a very detailed models. I'm going to take a different approach here and start showing you some extremely. It's in his diagram is the simplest possible models. It's just a very old model, which I'm very proud of. It's been 20 years now. Suppose you take a very simple model for neuron, just something like an excitable cellular automaton that spikes if sufficiently stimulated, and then it goes through a refractory period and goes back here. So this is a almost offensive cartoon of the Hodgkin-Huxley model, right? And imagine now you put these things here. So each one of these nodes here would be one of these guys, and you connect with some probabilities through which a spiking neuron here could excite this one here if it is quiescent here. And after they spike, they go refractory period. And you put these guys on some topology and see what happens, right? And you can characterize this kind of average coupling by some kind of local branching ratio, which is the sum of these PIJs here. This will be 20 years next year, this paper. So let me see if I can still do it. So imagine I have this, for simplicity, I put in 2 dimensions, but it could be any other topology, this 50 by 50 grid of very simple neurons. And here on this horizontal axis, I'm showing the stimulus in log scale, and in vertical axis, I'm showing the response. Here linear scale, and here log scale. They are the same plot, okay? And suppose this, what is a weak stimulus here? It's just a Poisson process, okay? So these neurons are not connected, no coupling. So a Poisson stimulus arrives, and the guy spikes. If I increase the stimulus, well, this happens more often. No? It happens more often. And therefore, this response here, although it's on log-log scale, this is a linear response, okay? So this is boring, right? And eventually, if you take a very strong stimulus, this thing eventually saturates here because you have some refractory period. So this is a very crude model of non-interacting neurons. If you add some coupling, like in those probabilities, you increase those probabilities, what happens now is that at each incoming stimulus, you generate some kind of evanescent excitable wave, which dies out after a while, and therefore your response here, your average response, so will have increased, right? And therefore, what we're seeing here, these excitable waves amount to some amplification, which then you can detect better because you have a better signal here arrive in your, suppose this is like the olfactory epithelium or your olfactory periphery, okay? Now, so this is good. So I can increase coupling. If I increase coupling even more, then I have more amplification. More amplification is good because I can feel better. So I'm open a bottle of vinegar here at the bottom of the room. The molecules diffuse and eventually bind to my olfactory receptors here, and I feel this is what is coming from the outside. This is what I'm feeling here, or sending to my brain, right? This is what my olfactory system is sending to the rest of the brain, to the cortex. So this is good. So more coupling, the better. More amplification. So it turns out that if you keep increasing the coupling, it will reach a point here. I take a very weak stimulus, which will reach a critical point of the system, in which even for very mild stimuli, you generate these waves which are on the verge of dying and not dying because you reach a critical point. If you increase coupling even more, so I'll claim in a minute that these critical excitable waves amount to optimal amplification, and I'll characterize that in a minute. If you increase it even more, you reach the supercritical state in which, even for very, again, weak stimuli, these waves won't never die again, right? So this means that if someone opened the bottle of vinegar at the back of the room, one molecule arrives at my nose, and I smell vinegar for the rest of my life. That's a very bad nose. Noses don't do that, right? We know. So this is too much amplification. It's like audio feedback when you're trying to you increase amplification with a microphone and they get audio feedback, and then this is bad, right? So you have this family of response curves, which for this very simple model, in a random graph where mean field theory works, we can do the calculations. As increase the coupling from here to here, let me introduce you why, in which sense this critical coupling could be optimal. You have the generic response curve here with some plateau here, some plateau here. Dynamic range is just the range of stimuli that you can appropriately code with this response function after you have discarded this plateau here and this plateau here. You take 10% here and 10% here and discard it because too close to the plateau, this function is not invertible, right? What your brain receives is this. What is incoming from the outside world is this. So if you put 10 to the minus 5, 10 to the minus 4 here, you cannot invert this function. This is bad coding. So it's just a very operational, very rough definition of how well a response function can code. And see what happens for this very simple model. So this upper plateau here is more, it just doesn't change much as you play with the coupling. And so let's fix this for a while. And now, so this is uncoupled neurons, zero coupling. You see the dynamic range is like this size, okay? So it's small. As soon as I start increasing the coupling, so it moves to the left and dynamic range increases. So I increase the coupling, dynamic range increases, etc., etc., and etc., up to the critical point. After reach the critical point, wait a second, because now, if you keep increasing, you have some self-sustained activity, which creates a new plateau, which you have to discount, because it's bad coding. Now, once you discount this plateau here, your curve is here, your dynamic range is smaller, actually. And if you keep increasing the volume of a loudspeaker as you speak in a microphone with audio feedback, it gets worse, as everybody knows. So it gets worse, dynamic range decreases, look here, here, and here, and here, right? So if I take this coupling and I looked at how dynamic range evolves as a function of coupling, you see that it increases up to the critical point and then decreases, right? So that if you make a plot of one against the other, you get a peak at here, the critical point. So this was published many years ago, and of course it's a very general property of excitable media. It can be reproduced by a number of different levels of neurons, levels of models, and etc. You can include inhibition and whatnot. And eventually, Osami Kinoyoshi and myself, we work on different models in which you can even work on the mechanisms, possible mechanisms which would tune the model here automatically. So, very nice. The question, of course, is it true? And some people have done experiments on this. I was very happy when this paper came out. I was happy for about 1 year, okay? Because someone cared to test his ideas in slices, and as soon as so most slices have large dynamic range, and as soon as disturbed with drugs, either to overexcite or over-inhibit the network, it gets worse. So this is very nice, but again, it's a cortical slice. A less reduced preparation would be somatosensory cortex. Again, anesthetized rats, well, data is not so beautiful here, but still has some kind of peak. So, cool. So this was 20 years ago. Eventually, we put together this electrophysiology lab and tried to revisit these ideas of criticality using other tools of statistical mechanics. So we were able to, we had these silicon probes in which you have, these are very dense electrodes here in silicon shanks, which we inserted in the primary visual cortex of anesthetized rats. So anesthetized with urethane. And this kind of anesthesia has an interesting property. This is like a couple of hours of experiment in which you have this coefficient of variation. It's just a standard deviation divided by the average of the population firing rate. So these are 3 raster plots from the same data set. So each line here is 1 neuron, each dot or line is just this one spike, and here I'm summing all the activity of all neurons. So here, this purple here corresponds to this purple here. So this is a very low coefficient of variation in the sense that this collectively, this population instantaneous firing rate has a small standard deviation as compared to the mean. And at the other extreme, something like this has a very large standard deviation as compared to the mean, and you have intermediate stuff like this guy, for instance. So this low CV and high CV corresponds to less synchronous and more synchronous activity. Synchronicity in this field has broad possibility of meanings. So when we talk to physicists, we have to be very careful to what we mean by synchronous. So I try to avoid the term as much as possible, but sometimes it's the word we use, right? So, so another kind of tool that we employed, again, in this literature of criticality, collective behavior, is to calculate avalanches. So we bin the data somehow appropriately. I won't go into the detail. We define avalanches as, an avalanche is anything that happens between two silences, given a specific bin, and we calculate the size distribution of an avalanche, so like the number of spikes here, and the duration distribution of an avalanche, so the how long does it take, how many bins did it last. And for some, depending on the value of CV, this function can be more or less well fit by power laws with exponents which not always agree with well-known universality classes. So, in fact, these exponents can vary continuously with this CV parameter with which we are parsing the data. And is there a criterion to choose which are the true ones? And in fact, the answer from rigorous statistical mechanics is yes, or so we thought that we could apply this to this data. And so, again, please, just to keep in mind that the idea is not to show all the details, but just the rough idea. So in statistical physics, we know that if we have something like this at a critical point, which we expect to be, then the average size increases as a function of duration with some non-trivial exponent. And we observe that as well. And this exponent also depends on how synchronous your activity is. Moreover, again, statistical mechanics, if the system is critical, then these exponents are not independent. They are supposed to satisfy scaling relation. And this is a much higher bar to pass. It's very hard to find a model which has this, and this, right? So I have 3 scaling laws plus a 4th one which connects the 3 of them. It's not so easy to find. In fact, we don't know any model that satisfies this thing which is not critical. So experimentally, the 2 sides of these equations are independently evaluated. So this comes from one kind of plot, these 2 comes from these other 2 kinds of plot. So the question is, since these exponents depend on how synchronous your data is on CV, and this guy as well, and this guy as well, if I plot the left-hand side of this equation as a function of CV, and the right side of this equation a function of CV, do they cross, right? And yes, they do. So different symbols here amount to different animals in an experiment, and this is a group data. So yes, they do have a specific value of CV, and moreover, this is the residence time, the group data. This is where you spend most of your time. So most of the time, the data is there around this critical point that we seem to have discovered here. And when we reproduce apply this to publicly available data, we get similar results. Anyway, so this is another technique that we employed. Now, putting together data analysis and experiments done in our own lab. Later, we revisited these results with a model, which I will not have time to show, and shown with a model that maybe we had overinterpreted these results here. Those who were in the school before this one, I talked about it in more detail. But again, the way I started this thing is that brain criticality should confer functional cognitive advantages to this putative brain. And so far, I told you about slices and anesthetized animals. So they're not doing anything. So what are the advantages? Can brain criticality be linked to enhanced performance in a task? And we claim that yes, we do. I'm not going to talk about our recordings, but recordings that we took from a much more powerful agent, actor here, which is Allen Brain Observatory. So they perform huge experiments with lots of animals, lots of data, and make it publicly available. It has changed the way people do those things because I will not compete with them. So I just take their data. So they have a setup in which they have mice, pre-trained mice with head fixed, and the task of the mice is to detect a change of images in a sequence. They have an image and another one, and then it can change. When the image changes, the mouse has to lick in order to be rewarded. If he licks in the wrong time, he doesn't get rewarded, right? And they have very dense neuropixel recordings of primary visual cortex with large number of neurons. So for 44 mice in 62 sections, it's a lot of data. It's a lot of data. I don't know any other lab which can compete with these guys in this kind of sheer size of the experiment. And the question is, how do we assess criticality in this data set? By then, we had realized that this avalanche analysis that I showed before has a lot of methodological issues. We were then experimenting, trying different techniques. So let me make a dramatic pause. Okay, let's talk about the renormalization group. So the renormalization group, let me introduce the Ising model, which we have spins up and down. They interact with their first neighbors here with some coupling J0. And eventually, so you can now take the average activity of blocks of spins and take like the majority vote, for instance, and do a super spins. And mathematically, you describe this system as the super spins by interacting with some coupling J1, which is a function of J0 and other parameters of the model. And then you take this thing. So, I'm sorry, what you're doing here, you're changing the scale at which you're looking at the system, right? So if you do this again, now you have J1, you go to the J2. So you're changing again the scale at which you're looking at the system. And you have a mapping of J0 to J1, et cetera, et cetera. And eventually, if this guy reaches a fixed point, by definition, you're reaching scaling variance, which is a key signature of all these power laws that I'm showing before. So this power laws signal scaling variance. You change the scale, the system doesn't change. So this is the cartoon picture. This is what you get from, if you read Michael Fisher's review 1998, in which you take model parameters here. So this external field is like temperature, coupling, and you see that this critical point here will eventually, with these transformations of a renormalization group, come to a fixed point, which for those in the know is just a saddle, which separates something which would be subcritical, like disorder, or supercritical, like superordered. And this guy analytically does this. This is the gold standard in theoretical physics for understanding phase transition models. And this guy here presents scaling variance and non-Gaussian distributions because you are in such a particular case. When you renormalize your data the way it was described, here you have non-trivial things. The thing is that in models, of course, with the exception that Enzo just showed us in the previous presentation, it's very hard to find good models of the brain. And so here comes Bill Bialek, good friend of this place. He's always here, and introduces the phenomenological renormalization group, which is a data-driven, model-free approach to this issue. So in his paper, he takes, for instance, this is calcium imaging from the hippocampus, I think, and what he does is recursively group neurons with their most correlated unit. So he replaces interaction with correlation, right? And just groups, and then groups again, and then the groups again. And observe whether observables scale on trivially with cluster size, and observe whether normalized activity, after you do this a number of times, converges to non-Gaussian. It was published in these two papers, 2017, 2019, and then a bunch of papers followed, including some from our own group. And so this is called real space phenomenological normalization group, and I'll just go very quickly so you have an idea. This is how non-Gaussian your activity can become, and they study things like silence probability in a number of spikes here. So minus log of the silence probability exponential, it goes with the exponential of minus a. The size of these clusters, size 1, size 2, size 4, cluster of neurons. So if beta equals 1, this is just a Poisson process, okay? Great, thank you. Variance of the sum activity. So if alpha equals 1, this is the central limit theorem. If alpha equals 2, this is just perfectly correlated variables. Both cases are trivial, but you obtain something in between here. Correlation time increases with some power z of the system size, and more complicated stuff like how you fit the eigenvalue spectrum of the covariance matrix. We're going to focus on this thing here, actually on a variant of this normalized activity. So we come back here, and so how do we assess criticality? Let me show you how the experiments work. They're recording V1, primary visual cortex of the mice. They have this 60 minutes period in which the mice are being presented with those images, in which they have to lick to get rewarded. And then another 25 minutes in which they are presented with non-natural images, Gabor patterns, stripes and stuff, and gray screens, stuff like this. And then they bring back the same set of images as here, except that the lick pore has been removed. So the poor animal is being images for which he has been trained for weeks, except that now he cannot be rewarded. It must be very frustrating, I can only imagine. Now, so what Gustavo did here, take this like 30-second windows of data and apply the momentum space version of the phenomenological renormalization group, in which you take eigenvectors of the covariance matrix and you project onto a subset of these eigenvectors, the subset with the largest eigenvalues, and see how this thing evolves. So, for instance, I take this one neuron, this blue neuron here, and so if I project into all the eigenvectors, I have the same thing as originally I had. If I take half the eigenvectors, it has some coarse graining, and then I do it more and more and more until the limit of, that's why I need many neurons. And this is the coarse graining procedures. If I do this for all the neurons and then look at the probability distribution of this activity at the end of the procedure, so n over 4, n over 8, n over 16, you see here you can have a highly non-Gaussian distribution at the end. So in line with this idea that's what you would expect at a critical point in the normalization group phenomenon scenario. So here I'm just showing the value of the kurtosis, which is a very, very simple crude indication of how non-Gaussian you are. For the Gaussian, you expect kurtosis of 3, I think. And if you shuffle the data, you do get a Gaussian kind of stuff, right? And of course, this is for one example of one window. But you have other windows in which things are not so non-trivial. Okay? So it can be non-trivial, it can be kind of trivial. The question is this difference enough? Is this sufficiently non-Gaussian? I'll come to that in a minute, right? So the brain of the animal is fluctuating across these different collective states. And so in the picture of the renormalization group there should be before, the theoretical one, what we try to understand is you have this fixed point here, and you have the trivial fixed points, this non-trivial fixed point, and the trivial fixed points away from that. What you have here is this blue one, some kind of coarse graining procedure which brings you perhaps closer to this non-Gaussian fixed point, and other windows in which the coarse graining procedure brings you further away from this non-trivial fixed point. And surrogate data certainly does that. So in a way, the idea here is that low kurtosis of the renormalized activity is here, and higher kurtosis means you're closer to this supposed non-Gaussian fixed point in such a way that kurtosis works as a kind of distance to triviality, right? I'm almost there. I'll be there in a minute. So how far must one be to be considered critical? You have the distribution of kurtosis of all the whole data, and this is the surrogate data. So you could be here, over-enthusiastic gullible, the z-scorer, the skeptic, the denier, right? So what we do is propose to give you a knob so you can choose however you want. And you put your threshold here, and whatever is below the threshold is considered no scaling for you, and whatever is above the threshold is considered scaling for you. You choose whatever you want, and now what we do is look at all those windows, scaling or no scaling, and within those windows, the mouse is performing, hitting and missing, hitting and missing, missing and hitting, hitting and missing. And so we control for this, we shuffle these hits and misses. Now the question is there, how does this into triviality? How, as you move to this axis, does that impact performance? The critical brain hypothesis will predict that yes. Is it true? And this is the most important plot. I think the hit rate is very high in general. So these mice are very overtrained. But as you increase kurtosis, so distant to triviality, so to speak, you have better performance. If you normalize the data to show relative performance gain, it goes like the typical case, like 10%, around it can be up to 24% in gain in performance, right? So just to wrap up, of course, I cannot do the performance here because the mouse cannot perform. But what I can show here is that this is the cumulative distribution of kurtosis. And here, once the leak port has been removed, even though we're measuring the primary visual cortex and the images are the same, the fraction of windows above a given kurtosis threshold is smaller. So we have kurtosis closer to surrogate values. And you bring it here, it's even worse. So that's the second idea here, that the signals criticality are modulated by task involvement and engagement. So this is recruited by the animal somehow, this regime. And this is a paper done by Gustavo Cambraia. I just want to finish that we would love to run this PRG on your model or data if you're interested. It's a very broad technique. These are a summary of the techniques we employ, of the experimental infrastructure we have available, and yes, we share data with our collaborations if you're interested. Thank you very much.
Thank you. Thank you, Mauro. I'm sorry to interrupt, but we are
that's
really
fine. It's
running
great.
late. So we have time for a few questions if you... We don't have any time. Okay.
Okay.
Okay, so...
Not okay?
Okay, so...